Can you start, ma'am?
Yeah, please.
ಸೊ ಎಲ್ಲರಿಗೂ ನಮಸ್ಕಾರಗಳು. ಜೊತೆಗೆ ಎಲ್ಲರಿಗೂ ಶ್ರೀಕೃಷ್ಣ ಜನ್ಮಾಷ್ಟಮಿ ಶುಭಾಶಯಗಳು ಮೇಡಮ್. ಹಲೋ ಹಲೋ
We can hear you.
ಮೇಡಮ್, ತಮ್ಮ ಬಗ್ಗೆ ಒಂದು ಕಿರು ಪರಿಚಯ ಮೇಡಮ್.
It was not needed.
Okay, just I give a short introduction about you.
Okay.
So Dr. Jyoti madam completed his MSc Biotechnology in 1998, awarded MPhil in 2008. From Bharati Darshan University and a PhD in 2014 from Manipal University for thesis titled on study of in vitro and in vivo antioxidant and anti-diabetic activities of cassia areata, and the guided student research project led in crayons funded by National Reference Center for Lead Poisoning in India, Bangalore and Surana College in 2012. Study of in vitro antioxidant and antimicrobial activities of cassia papaya leaves spice project sanctioned by Government of Karnataka Vision Group on Science and Technology VGST in 2014-2015, and currently guiding a student in house research project synthesis and characterization of ZNO nanoparticles from Moringa oleifera flower extract at BMSCW, and 5 research publications in peer-reviewed international research journals and 2 paper presentation and 2 poster presentation in national conferences. Also, the co-authored book chapter titled The Recent Precipitation into Extraction of Nanocellulose Crosstalk between Natural Resources and Progressive Applications for Handbook on Natural Polymers. Publishers Elsevier has been accepted number 65563, and it is under the process of publications. Served as BOS member under Bangalore City University 2019-2020, and currently BOE member Biochemistry section 2021 to 22. Presently, Madam working as Assistant Professor, Department of Chemistry in BMS College, Bangalore. Okay, thank you, ma'am. Over to you, ma'am.
Thank you so much, Shri Siddegowda, the curator. At the outset, I would like to thank KSTA for inviting me to give a guest lecture to the students of second PUC, those who have interest in science. And I appreciate the initiative taken by the KSTA in, supporting the or building up or inculcating the scientific temper in the students. I also thank Dr. A.M. Ramesh, CEO of KSTA, for inviting me to give the lecture. Thank you so much. And I would like to say namaste to all the students. I see very hardly some students who are present there. And Makle, I hope that you will be able to understand some of the topics which are very relative to you, which is there in your syllabus. If you have any problems, you can always send the message in the chat box so that we will be able to help you out with your problems. Shall we start? If you say, you can give a thumbs up. Wherever you are going to be having a problem, you can just, interrupt in between by, maybe you're allowed to only send the messages in the chat box, I believe. Shall we start, Makle?
Yes, start, ma'am.
Okay. Right. No, is this screen full? Full screen ಬರ್ತಾ ಇದೆಯಾ?
Yes, madam.
Okay, then I think ನನಗೆ problem ಆಗ್ತಿದೆ. Okay, no problem. Starting with what is called as the biomolecules.
Just click
Bio.
the up and arrow mark.
ಕೇಳಿಸ್ತಿಲ್ಲ, ಏನ್ ಹೇಳ್ತೀರಾ?
You just change the ಇದನ್ನ change ಮಾಡಿ ಮೇಡಮ್, PPT ನ change ಮಾಡಿ, ಅದು change ಆಗುತ್ತೆ ಏನು ನೋಡಿ.
ಚೇಂಜ್ ಆಗ್ತಾ ಇದೆ.
Yes madam.
ಬಟ್ ಫುಲ್ ಸ್ಕ್ರೀನ್ ನನಗೆ ಕಾಣಿಸ್ತಾ ಇಲ್ಲ, ಬಟ್ ನಿಮಗೆ ಕಾಣಿಸ್ತಿದ್ರೆ ಐ ಹ್ಯಾವ್ ನೋ
ಇಲ್ಲ
ಪ್ರಾಬ್ಲಮ್.
ಇಲ್ಲ, ಮೇಲ್ಗಡೆ ನೀವು ಫುಲ್ ಸ್ಕ್ರೀನ್ ಇದೆಯಲ್ಲ, ಕ್ಲಿಕ್ ಮಾಡಿ ಮೇಡಮ್ ಅದು.
Okay, I'm not getting a slide show. Having little problem. Okay. Now it is okay.
Yes, now it's full screen ma'am.
Okay. Makle, we will start. Biomolecules. Bio means living. Molecules means the compounds which are going to be present within the living system here. So they are going to be involving a wide range of molecules which are going to be differing with respect to the functional groups, with respect to the size, the shape, as well as they are expressing a vast array of the functions. Okay, and today I am going to be giving you a little bit of an overview of the carbohydrates and the proteins here. Both are very abundant in nature. ನಿಮಗೆ ನೇಚರ್ ಅಲ್ಲಿ ತುಂಬಾ ಕಾಣಿಸುತ್ತೆ ಇದು. They are available in nature. Plus, with the apart from being present in the nature, they are also going to be having a especially with respect to the proteins, they are going to be highly complex and showing again very vast functions. Let's come to the carbohydrates here. The definition of the carbohydrate is they are going to be what is called as the polyhydroxy aldehydes or polyhydroxy ketones here. Poly means many, hydroxy means containing an OH group, aldehydes means containing the functional group CHO. Whenever they are going to be having an aldehyde group, the carbohydrates will be considered as an aldose. Whereas if they are going to be having the functional group, apart from having the polyhydroxy group, if they contain the C double bond O group, which is nothing but a carbonyl group, they are considered as ketones, polyhydroxy ketones, which may also be known or called by ketosis here, represented by the general formula CnH2ON. So the number of the carbon atom with respect to the water molecule present is going to be the same here. Okay, that's why it is going to be represented as Cn and H2O n times here. So when we are going to be giving the definition of the carbohydrates, you can also, include a word called as optically active polyhydroxyaldehydes and optically active polyhydroxy ketones. If you write this, you will be more precise. ನೀವು ತುಂಬಾ ಕರೆಕ್ಟ್ ಆಗಿ ಇನ್ನು ಜಾಸ್ತಿ ಅದ್ರ ಬಗ್ಗೆ ಹೇಳ್ತೀರಾ ಅಂತ ಗೊತ್ತಾಗುತ್ತೆ. What do you mean? What is the meaning of the optically active here? Optically means the interaction with the light. They are the compounds which have the capacity to interact with the light. And what happens when the light is going to be passed through them? Not the normal light, but it is going to be a plain polarized light. I think you can make out in this particular diagram here, which is showing the normal light going through the filter, and that filter is going to be releasing only a light which is going to be having the vibration in only one particular direction here. So that is what is called as the plain polarized light. And that plain polarized light, when you are going to be making it to fall on a compound here, for example, any carbohydrate for that matter, they are going to be bending that light either towards the right or towards the left here. That's why it is called as optically active. Okay. And coming to the classification of the carbohydrate, it is classified as monosaccharides, oligosaccharides, and polysaccharides. Monosaccharides, these are again, the compounds which are going to be having the polyhydroxy groups with the major functional group, which is nothing but the aldehyde or the ketone. or on hydrolysis, which is going to be, obtaining, or which is going to be forming such compounds here. So this cannot be further can be, hydrolyzed. Monosaccharides are actually the compounds which further cannot be hydrolyzed. So they are going to be involving it starts from trisaccharides. So 3 carbon sugar, 4 carbon sugar, 5 carbon, 6 carbon, like that. Okay. So some of the examples are going to be, glucose comes under the monosaccharides, as well as you will have what is called as the ribose, which is a 5 carbon compound. You will also will be having a 3 carbon, but the glyceraldehyde is an example for the monosaccharide. Coming to the oligo means it is going to be somewhere varying with respect to 2 to 10 monosaccharide units can be obtained when such oligosaccharides are undergoing the hydrolysis. So the best example or the common example which you can observe are going to be the sucrose here. Sucrose is a very common, disaccharide. Along with that, you will also know the lactose, which is a milk sugar, as well as the maltose, starch nalli irutte. Raffinose is actually is a what is called as the trisaccharides here. Tri means more sugar molecules have been joined with respect to each other to give rise to a trisaccharide named as raffinose. Raffinose composition is going to be the galactose, the fructose joined through the glucose molecules here. Coming to the polysaccharide, polysaccharides are going to be the larger molecule. The bulk molecules are the hierarchy. In terms of hierarchy, they are going to be the the compounds with many number of hundreds and thousand number of the sugar units which have been joined together with respect to each other, brought together to give rise to a complex compound, and which is going to be available in nature. And the most abundant polysaccharide which exists in the nature is going to be nothing but the cellulose, which is going to be seen in case of the plants here. And along with that, you will also will be having what is called as the glycogen, as well as the starch here. Starch and the glycogen are going to be the storage form of the food here. Okay. Then coming to directly into what is called as the elucidation of the structure of the glucose here. If you understand the elucidation and the present structure of the glucose, which you have observed or which you always will be writing while depicting. If somebody is going to ask you, write the structure of the glucose, how are you going to write that? And to write that particular structure, you should have, certain support for that. And that support, there many scientists have worked endlessly years together to arrive at that particular structure of the glucose, and which you are going to be studying under the elucidation, how you arrived at the structure of the glucose. So according to the, you will start with the elementary analysis. Elementary means elements. ಯಾವ್ಯಾವುದು atoms ಇದೆ ಅಲ್ಲಿ. What are the elements present? Carbon is there, hydrogen is there, oxygen ಇದೆ ಅಂತ ಹೇಳ್ಬಿಟ್ಟು elementary analysis ಇಂದ ನಿಮಗೆ ಗೊತ್ತಾಗುತ್ತೆ. So elementary analysis as well as the molecular weight analysis ಅಥವಾ molecular weight determination has, with these analysis, you can make out the molecular formula to be C6H12 and O6. That was the first actually what is called as the data which they could obtain here. So when they started, if that is going to be the molecular weight and it is going to be having 6 carbon, 12 hydrogen and 6 oxygen, and how you have to arrange them in the space here, how they are connected with respect to each other. So to that particular compound, so when you are treating them with the hydroiodic acid, so I think you can see this particular reaction. This particular reaction is going to be representing the reaction of the glucose with the hydroiodic acid. It's a continuous reaction or a prolonged reaction, prolonged treatment of the glucose with the HI with the red phosphorus, giving you what is called as the N-hexane here. So this N-hexane, which is the product, it indicated the presence of these 6 carbon in a single chain here. So you can make out this 6 carbon present in the single chain forms the backbone of this carbohydrate or the sugar. Next, what they did is that, okay, if this is going to be the case, they treated the glucose with the HCN molecule, hydrogen cyanide molecule. The product formed is nothing but cyanohydrin. Cyanohydrin. This is actually is going to be an adduct. What is an adduct? The hydrogen is forming a bond with the oxygen and the CN is been joined to the carbon here. So it is just the addition of the hydrogen cyanide to the glucose giving rise to the glucose cyanohydrin here, which is an adduct form. And plus, they also treated the glucose with the hydroxylamine. Hydroxyl means OH, amine means NH2. So NH2 OH is going to be treated with the glucose to get what is called as an oxime here. So what is that oxime here? This oxime is nothing but the condensation product of the glucose with the hydroxylamine. So what is the inference of the reaction of the HCN as well as the hydroxylamine with respect to the glucose molecule? So which is going to be nothing but the representing the presence of the carbonyl group. Okay, so the carbonyl group is going to be present, but they were unable to tell, these two reactions were unable to tell whether the compound is going to be, aldehyde or the ketone. So to find out whether the compound is going to be having an aldehyde group or the ketonic group, they treated the glucose with respect to the mild oxidizing agent, which is nothing but the bromine water here. So this particular bromine water oxidize the aldehyde to give rise to COOH, conversion of an CHO into a COOH, representing that the carbonyl group is nothing but attached to the hydrogen, which means that the glucose contains an aldehydic group. Next, coming to, if the functional group is going to be the aldehyde, what about the remaining, the attachment of the hydrogen, oxygen, and the carbon here? So for that, they actually treated the glucose with the strong oxidizing agent here. So the strong oxidizing agent not only oxidize the aldehyde group, but remaining, the sixth carbon, which is going to be containing a primary alcoholic group, is also going to be, oxidized here. So mild oxidizing agent is going to be converting an aldehyde into an carboxylic acid, whereas the strong oxidizing agent, which is nothing but dilute nitric acid, is oxidizing the aldehyde as well as the primary alcohol into respective carboxylic acid here. Okay, so indicating the presence of the not only aldehyde group but also the primary alcoholic group here. So when they treated the glucose with the acetic anhydride here, they formed penta acetate derivative of the glucose. Okay, so this particular product represented, there are actually are going to be 5 hydroxyl group present. Out of 5 hydroxyl group, one is going to be the primary hydroxyl group, just as you have seen from the previous reaction, and the remaining with respect to the penta-acetate derivative here. So based on all these reactions, a lot of research was done on, these are the traditional ways to find out the, presence of the functional group, which is going to be the attachment, where actually they are going to be present. So based on these results, they could arrive at the structure. They actually proposed an open chain structure, which is going to be represented like this here. So this is conventionally is going to be written as, the first carbon is going to be the always is represented by the functional group here. So having an aldehyde group, and the remaining are going to be, you will mark it as plus, minus, plus, and plus here. Plus representing the presence of the OH group on the right hand side. Minus means OH group present on the left hand side here. Okay, so here the sixth carbon is going to be the carbon which with the the primary alcoholic group. Okay. Hope you are following, Makle. Can somebody give me a thumbs up? Can you raise the hand? Are you following? Okay, I'm unable to see that. Now, based on that, the previous findings, with the help of which they were able to arrive at the open chain structure of the glucose, but later on they found that the typical reactions of the aldehyde. So the typical reactions of the aldehydes are nothing but forming an adduct with the sodium bisulfite, one of the very important reactions of the all the aldehydes here. And along with that, they are supposed to give what is called as the pink coloration with the Schiff's base. Okay, so both these are not going to be answered by the glucose molecule. So apart from that, the glucose pentaacetate compound, which is going to be formed when you are going to treat a glucose with the acetic acid, also is not going to be reacting with the hydroxylamine. We have seen in the previous case the hydroxylamine, which is going to be forming what is called as an oxime here. This particular oxime formation is exclusively only involving the aldehyde group. This is also is not going to be shown. So what is the reason for that particular rigidity? Although individually they have given all these shown reaction, but whenever there is going to be they treated it with the what is called as sodium bisulfite as well as the Schiff's base, they were unable to get the common reactions of the aldehydes. Find it. Okay. Coming back to this, so what is the reason for that? And later they could found out, what they could found out is that if you're going to be observing this particular what is called as the slide representing the first carbon having a kind of a interaction with the fifth carbon here. How? Now this particular CHO bond, if you are going to be observing, is forming a bond with the hydrogen of the fifth carbon here. So what exactly is happening here is that the aldehyde group is forming a bond with the OH group of the fifth carbon here, forming what is called as the hemiacetal here. So this is very clear here. So hemiacetal, what do you mean by the hemiacetal here? Hemiacetal is nothing but forming a kind of, this is going to be carbonyl group. This carbonyl group is going to be, because it is going to be polar in nature, the pi bond is going to be shifted onto the oxygen because of the negative charge being negative here, carrying a negative charge. So becomes C single bond O minus, which is going to be attracting the H plus H ions of the fifth carbon here. Okay. So what happens? This C double bond O is going to become COH. And because this particular carbon has lost this hydrogen, it is going to be joined, it is going to compensate that with joining with the carbon here to give rise to a closed ring structure, which is as equal to, or it is equivalent to what is called as the glucopyranose here. So gluco means glucose, pyranose means, so it is going to be almost similar to the structure of the pyran ring. Pyran ring is a heterocyclic molecule here, heterocyclic molecule containing the 1, 2, 3, 4 and 5 carbon, and the sixth atom present is going to be the oxygen. Okay, so this is actually is going to be the representing the anomeric glucose. Then what do you mean by the anomeric here? There are two anomeric forms of the glucose. One, when the OH group is going to be present on the upper side. Okay, so remaining other, you can remember it like this, the OH group like this, and this OH group like this. So here representing the beta glucopyranose structure here. If the OH group is going to be present downward here, then it is going to be considered as alpha D glucopyranose here. So this is the Harvard projection of the structure of the glucose here. Then coming to the structure of the fructose here. Fructose is also, they are isomers of each other, sharing the same molecular formula. This same molecular formula, but containing a different functional group, it is nothing but a ketone here. So here also the fructose is going to be represented through a 5-membered ring, where it is going to be showing the most stabilized configuration here, considered as the fructofuranose here, because it is going to be representing the structure of the furan. Furan is a heterocyclic compound, just like the pyron here. So this is also going to be representing, anomer, where the OH group is going to be present on the upside. You will have a beta structure. If it is going to be present on the downward, you will consider it as an alpha fructopyranose. So this particular glucose is going to be levorotatory. What do you mean by levorotatory? Levorotatory is when the plane of the polarized light is going to be passed through the fructose molecule. The light is going to be bent towards the left side here. That's why you call them as levorotatory. If the light is going to be, bent towards the right, the compound which is going to bend the plane polarized light towards the right, you call them as dextrorotatory, and the glucose is going to be dextrorotatory in nature here. Then coming to what is called as the glycosidic linkages here. So glycosidic. So how do you get this glycosidic linkages? You observe this glycosidic linkages in the disaccharides here. So what are disaccharides? Disaccharides means, di means two, saccharides means sugar. So two sugar molecule are present in a molecule here, which is going to be considered under the oligosaccharides here. So one of the most common, example of the disaccharide is going to be the maltose here. Maltose, two glucose molecules are going to be brought with respect to each other through the condensation. So condensation means you are bringing two compounds together with the removal of some compounds here. So what is going to be removing here? See, the OH group of the one glucose molecule is going to react with the OH group of the another glucose molecule, and the water molecule is going to be removed. OH plus OH. If you remove water molecule, the one oxygen is going to be remaining, which is going to be connecting this first glucose with respect to the second glucose molecule. And that's why you are going to be considering this as the glycosidic linkages. And how do you represent them? It is going to be alpha because it is going to be present downward. Alpha D, capital D is representing the configuration. Configuration is the arrangement of the hydrogen or the arrangement of the atoms in a 3D space here. Alpha D, gluco means glucose. Pyranose means it has a pyranose ring structure here, which has formed a glycosidic bond with another glucose molecule. So it can also be, you can have the beta linkages also. This is alpha linkage. Beta linkage means when the OH group is going to be present above the plane here, which is going to be again involved in the glycosidic linkages, but it appears like this. And because of the glycosidic linkages, usually they are going to be a kind of showing and chemically inert with respect to, they are chemically inert means they show resistance to the reactions here, or they may show resistance to the action of the acids or the bases here. They are little, strong when compared to the alpha linkages here. Coming to what is called as the structure of the sucrose molecules here. So sucrose is the most commonly found, sugar molecule, which is a disaccharide, which is going to be most abundantly found, seen in case of the fruits. And this is going to be involving a combination of the glucose as well as the fructose here. So when you are going to be observing this particular structure of the sucrose here, the anomeric OH group, anomeric carbon is going to be involved. Anomeric means basically it was an aldehyde group. Then later on it switched, or it formed a hemiacetal, and that containing an OH group is going to be involved in the bond formation here. So why you have to have this in your mind is that if the sugar molecule is having a free OH group, just like here, I will just show it to you. This is going to be showing a free OH group here. Here, it has been involved in the glycosidic linkage. So here, it is involved in the glycosidic linkage. But here, you can observe the presence of the OH group, which is an anomeric, what is called as the OH group present on the anomeric carbon. If they are free, they are going to be acting as reducing agents here. So very important. So here, if you are going to be observing the structure of the sucrose molecule, you don't see that, anomeric carbon having a free OH group. And that's why the sucrose is going to be non-reducing in nature here. So there are actually certain tests are present here, with the help of which you can make out whether the compound is going to be carbohydrate or not, if it is going to be carbohydrate, whether it is going to be reducing or non-reducing, and if it is going to be carbohydrate, whether it is going to be an aldose or a ketose here. Okay, so Molish test is going to be the test which is going to be answered by almost all the carbohydrates here. So in this particular test, you are going to be taking the carbohydrate, you are adding the beta naphthol to that, along with the presence, addition of an acid here. So acid is going to, dehydrate them. So this to form an aldehyde, this aldehyde reacts with the phenol to give rise to the purple color ring at the junction of two layers here. Very, definite answer, very conspicuous by its presence, only answered if they are going to be, carbohydrate in nature. So along with that, if you are going to be taking the sugar molecule, you're adding a Benedict's solution. So what is Benedict's? Benedict's is going to be nothing but a combination of hydrated copper sulfate with the in presence of a sodium carbonate and sodium citrate. So when you are going to treat them and then heat them, if you are going to be getting an, dark orange colored precipitation or dark brown color precipitation, so it shows the presence of the reducing sugar. So what do you mean by reducing sugar? Those where the OH group is going to be anomeric carbon having a free OH group. If they are present, they are going to be reducing in nature. Then coming to the Selvinov's test, it is the test to indicate the presence of the ketones here. If the if the fructose is going to be present, if xylulose is going to be present, so naturally they are going to be the carbohydrate containing the ketone group here, C double bond O group. So here they are going to be answering the test, and by representing or by showing a cherry red coloration, and you can distinguish between an aldohexoses or aldoses or ketosis. Okay. Then coming to what is called as the starch here. So the starch is going to be the polysaccharide. Poly means many number of the molecule, monomer units or the glucose units are coming close together with respect to definitely the glycosidic linkages here. Glycosidic means, so each sugar molecules are going to be joined, and during the joining, the water molecule is going to be removed here, which you call it as a condensation reaction here, and they are joined through alpha 1 to 4 linkages here. So this is going to be representing alpha 1 to 4 linkages here. So very neatly they are actually been, joined with respect to each other. This is a partial, structure of the, starch here. And this particular starch is a combination of amylose and amylopectin. So amylose is going to be somewhere around, they constitute about 15 to 20%. Starch ನಲ್ಲಿ 15ರಿಂದ 20% amylose ಇರುತ್ತೆ, ಉಳಿದಿದೆ 80% amylopectin ಇರುತ್ತೆ. So what are amylopectin here? So this amylopectin is going to be having a branched structure. So ಇಲ್ಲಿ ನಿಮಗೆ linear structure ಬಂದಿದೆ, linear structure. Here you are going to be having a branch structure. So how do you get this branches? And when the, different, all this, this is one layer or linear layer of the what is called as the amylose, and this is another layer of the amylase, and these two amylose, the what is called as the layers are, I mean, the units are going to be joined through the 1 to 6 linkage. This is the carbon number 1 anomeric OH group is going to be involved in forming a bond with the 6th carbon of the next, polymer here, polymer unit, which is lying next here. So they are highly branched here. And starch is going to be involving, these are the storage polysaccharides, storage food in case of the plants. So then coming to what is called as the cellulose here. Cellulose, as I told you, this is going to be the most abundant, as I told you at the beginning of the class here. And here the difference between the uh, starch and the cellulose, if you observe them, the linkages is going to be involving the beta linkage. I mean the OH group which is present above the plane is going to be involved here, whereas in the alpha linkages, the OH group which is present below the plane are going to be involved in the glycosidic linkages here. So this is going to be, this cellulose is going to be seen in case of the plants. So they are going to be the components of the cell wall here. As I told you, they are the most abundant, the carbohydrate in biosphere actually. And as I told you, they are chemically inactive in nature. I told you they are going to be resistance to the action of the acids, the bases, or the oxidizing agent and the reducing agent. And this actually is very important for the usage of the cellulose here, which we are going to see it in the next slide here. Then coming to the, the structure of the glycogen here. This is called as the animal starch. So because it is going to be seen in case of the animal, this the storage form of the food in case of the animals here. Not only, you can find them in case of the muscles or in case of the livers here. And it is almost, similar to the starch, but the difference here is that it is highly branched. Okay. Then coming to the last slide of this particular, the carbohydrate slides, the importance is they act as biofuels. So what do you mean by the biofuels here? So biofuels are going to be acting, they have the calorific value. Calorific value ಅಂದ್ರೆ ನೀವು ಅದನ್ನ ಕನ್ಸ್ಯೂಮ್ ಮಾಡಿದ್ರೆ ಫುಡ್ ನಲ್ಲಿ ನೀವು ತಗೊಂಡ್ರೆ ಕಾರ್ಬೋಹೈಡ್ರೇಟ್ ನ, it is going to be giving you a lot of energy. Nearly 55 to 65% of the energy daily intake of the food ನಲ್ಲಿ ನೀವು ಏನ್ ಕಾರ್ಬೋಹೈಡ್ರೇಟ್ ತಗೊಂತೀರಾ, ನಿಮ್ಗೆ ಏನ್ ಎನರ್ಜಿ ಬೇಕು ಅದು ನಿಮಗೆ ಕಾರ್ಬೋಹೈಡ್ರೇಟ್ ಇಂದ ಸಿಗುತ್ತೆ. ಅದಕ್ಕೆ ಅದನ್ನ ಬಯೋಫ್ಯೂಯಲ್, ಫ್ಯೂಯಲ್ ಮೀನ್ಸ್ ವೆನ್ ಯು ಆರ್ ಗೋಯಿಂಗ್ ಟು ಬರ್ನ್ ದಮ್ ಯು ಆರ್ ಗೋಯಿಂಗ್ ಟು ಗೆಟ್ ದಿ ಎನರ್ಜಿ. ಅದಕ್ಕೆ ಅದನ್ನ ಫ್ಯೂಯಲ್ಸ್ ಅಂತ ಕರೀತಾರೆ. ಓಕೆ, ಸೊ ಕಂಪೇರ್ ಟು ದಿ lipids here, fat is also, energy giving in nature, but fat energy not always you can accept it. There are only, uh, most commonly you get the energy from the oxidation of the carbohydrates here. So biological importance, very very important actually. They have a very high calorific value and they are going to be energy givers. So normal functioning of the body ge carbohydrates are very very important. But at the same time, one point ನಾನು ಇಲ್ಲಿ ಮೆನ್ಷನ್ ಮಾಡ್ಲಿಕ್ಕೆ ಇಷ್ಟಪಡ್ತೀನಿ, especially with respect to the cancer patients here. So cancer patients, and what is the meaning of that? So where there is going to be an, uncontrolled division of the cells are taking place here. So that is again energy demanding. ಆ ಪ್ರೋಸೆಸ್ ಮಾಡ್ಲಿಕ್ಕೆ ಎನರ್ಜಿ ಬೇಕು. ಆ ಎನರ್ಜಿ ಯಾರು ಕೊಡ್ತಾರೆ? Definitely again it is going to be given by the carbohydrates. So naturally ಕಾರ್ಬೋಹೈಡ್ರೇಟ್ಸ್ ಹೇಗೆ ನಮಗೆ ನಾರ್ಮಲ್ ಫಂಕ್ಷನ್ಗೆ ಎನರ್ಜಿ ಕೊಡುತ್ತೆ, ಹಾಗೇನೆ ಅದನ್ನ ಎಷ್ಟು ಬೇಕೋ ಅಷ್ಟು ಮಾತ್ರ ತಗೊಳ್ಬೇಕು ಅನ್ನೋದನ್ನ ಈ ಪರ್ಟಿಕ್ಯುಲರ್, ಎವ್ರಿ ಮಾಲಿಕ್ಯೂಲ್ ವಿಲ್ ಹ್ಯಾವ್ ಇಟ್ ಓನ್ what is called as an importance. But if you go on taking more and more of such, if you are going to feed on if you're going to feed on the excessively on the carbohydrate, naturally it is going to be leading to what is called as the mm, leading to the deleterious effects on the body. Okay, coming back to, as I was mentioning you, these are going to be acting as the constituents of the cell membrane. So cell membrane components held in cellulose and bilbitu. Then very, very important, they are also are going to be the components of the nucleic acids, especially the RNA as well as the DNA. Okay, so deoxyribonucleic acid is going to be the it is the carbohydrate found in case of the DNA, and the ribose sugar are going to be seen in case of the RNA molecules. Okay, so cellulose, non-halide, because they are going to be chemically inert, they are going to be used, widely used in the furniture industry as well as the cloth industry. Many other functions are also going to be that. And hope you have followed it. And here are some of the questions which you can answer. My cursor is not moving. Okay, so this is some of the questions which you can definitely answer. The number of the optical isomers of the glucose and the fructose are going to be. How do you find them? So just by calculating using the formula 2 to the power of n. N represent number of the chiral center. What do you mean by the chiral center? When the carbon is going to be linked to 4 different groups, you call them as chiral centers. And how many glucose, in a glucose molecule, there are 6 carbons are there. Out of that 6, there are actually 4 carbons are going to be surrounded by different groups. You can do it at home, definitely. So when you're going to substitute them, you will get the answer 60. And with respect to the fructose, there are actually 8 optical isomers are there. Raffinose, I told you it's a trisaccharide, and the diabetes is going to be detected by the Benedict solution. Levulose is going to be it's a name for the fructose molecule because it is going to be bending the light towards the left. That's why it is called as levorotatory levulose. Fructose gives a silver mirror test as well as the Fehling solution test because of enol formation. Enol means when the OH, C double bond O is going to be converted into COH group. It is what is called as the keto enol formation. Tautomerism ನಲ್ಲಿ ನಿಮಗೆ ಬರುತ್ತೆ. What is the value of the optical rotation of the invert sugar, which I didn't tell you actually. So sugar is, sucrose is going to be optically active and it is going to be dextrorotatory. It bends the light towards the right here. But on hydrolysis, sucrose will break down into glucose and the fructose here. And glucose is going to be dextrorotatory, fructose is going to be levorotatory. And since more bending is seen with respect to the fructose, since it bends towards the left, then that's why it is called as an invert sugar. Okay, then coming to the proteins here, I think I have left with another 10 more minutes, I'll be able to complete this proteins, I feel. So proteins are highly complex because they are going to be having the bifunctional group. What do you mean by bifunctional group? They not only are going to be having the COOH group, but at the same time, they're going to be having an amino group here. And since this NH3 group as well as the COOH group is attached to the same carbon here, and this carbon is considered as an alpha hydrogen because the primary priority should be given to the functional group and the priority is the carboxyl group. The next carbon is considered as alpha, and that's why they are alpha amino. They are named as alpha amino acids here. So based on the nature of the R group here, based on the nature of the R groups are going to be nothing but the side chain here. The amino acids are going to be categorized as the neutral amino acid, the positively charged amino acids, and negatively charged amino acids here. So negatively charged amino acids are going to be aspartate and glutamate, very easy to remember, whereas the positively charged amino acids are going to be lysine and arginine. Then the neutral amino acids are going to be serine, you can write, which is hydroxyl group containing amino acid, threonine, cysteine, asparagine, and glutamine here. Then based on the nutritional value, it's very easy to remember essential amino acid and non-essential amino acids here. Essential means the body requires them and the body is not capable of, synthesizing them within the system or within the body here. That's why you have to take it through the diet, because they are very important for the biosynthetic reactions here. So how you can, remember them? MAT will fly. MATT, MAT, VIL, will fly. How do you represent them? MAT means methionine, arginine, threonine, and tryptophan. V, valine. IL means isoleucine as well as leucine. PH means phenyl, phenyl alanine. H is histidine. LY is lysine here. So it is easy to remember for you. These are the essential amino acid as well as non-essential amino acid. So because of the presence of the bifunctional group, they are going to be representing what is called as a Zwitter ionic form. What do you mean by Zwitter ions here? Zwitter ions is the condition where they are charged, but they are going to be neutral. So even they are going to be charged means they are going to be carrying both negative as well as the positive charge, which get compensated with respect to each other, and hence they are going to be electrically neutral here. But the possessing positive and the negative charge at a time gives it a Zwitter ionic form here. Now how? Because within the physiological pH, physiological and body ಒಳಗಡೆ aqueous condition ಇರೋದ್ರಿಂದ ನೀರಿನ ಜೊತೆ ರಿಯಾಕ್ಟ್ ಆದಾಗ ಅಮೈನೋ ಆಸಿಡ್ ಏನಾಗುತ್ತೆ, ಸೊ ದ ಪ್ರೋಟಾನ್ ಇಸ್ ಗೋಯಿಂಗ್ ಟು ಬಿ ಡೊನೇಟೆಡ್ ಹಿಯರ್. ಆ ಪ್ರೋಟಾನ್ ಹೊರಗಡೆ ಹೋಗುತ್ತೆ, COOH ಗ್ರೂಪ್ ಇದ್ದಿದ್ದು COO ಮೈನಸ್ ಆಗುತ್ತೆ. ಆ ಪ್ರೋಟಾನ್ ಅನ್ನ NH2 ಇದ್ದಿದ್ದು ಅಕ್ಸೆಪ್ಟ್ ಮಾಡಿ NH3 ಪ್ಲಸ್ ಆಗಿ ಕನ್ವರ್ಟ್ ಆಗುತ್ತೆ. Okay, so they amino acids are, they act as not only proton donor as well as the proton acceptor. That's why they are called as ampholytes. Okay, amphifites antanu adana karitare. So hege, so in the acidic medium andre which are rich in H protons irutte. So proton jote react adaga enagutte, so they are going to be, can you make out, in the acidic medium they accept the proton, accept yaar ಮಾಡ್ತಾರೆ, they accept, proton is going to be accepted here, it becomes COH, so naturally they are going to be carrying positive charges. So in the basic medium, so they are going to donate the proton. So ಹೇಗೆ donate ಮಾಡುತ್ತೆ, NH3 plus ಇಂದ ಒಂದು proton ಹೊರಗಡೆ ಹೋಗುತ್ತೆ, it becomes NH2, so it is going to be carrying COO minus, they are going to be carrying the negative charge. Okay, so okay, this is going to be the slide representing the formation of the peptide bonds here. So peptide, we have seen glycosidic bond. Glycosidic bonds will get two different monomer units together with respect to carbohydrate. In case of the proteins here, so amino acids are going to be the building blocks. So ಹೇಗೆ ಇಟ್ಟಿಗೆ ಇಟ್ಟು ಒಂದು ಬಾಲ್ ಮಾಡ್ತೀವಿ, ಅದೇ ತರ ಇಲ್ಲಿ amino acid are going to be joined with respect to each other. How they are going to be brought with respect to each other? Through the formation of a peptide bond. ಹೇಗೆ peptide bond ಆಗುತ್ತೆ? First amino acid, the COOH group reacts with next amino acid, the amino group NH2 group ಜೊತೆ reaction ಆದಾಗ water molecule remove ಆಗುತ್ತೆ. Water molecule remove ಆದರೆ ಅದನ್ನ condensation reaction ಅಂತ ಕರೀತಾರೆ. The bond formed is going to be C double bond ONH, ಅದನ್ನ amide bond ಅಂತ ಕರೀತಾರೆ. Amide bond, especially in case of the protein, it is going to be called as a peptide bond. So ಇಲ್ಲಿ ಒಂದು amino acid has reacted with the next amino acid. So first amino acid, this is second amino acid. So the form agiro molecule na dipeptide anta karitare. Di means ಎರಡು amino acid join ಆಗಿ peptide bond through join ಆಗಿರೋದ್ರಿಂದ ಅದನ್ನ dipeptide ಅಂತ ಹೇಳ್ಬಿಟ್ಟು ಕರೀತಾರೆ. So coming to the different levels of the organization, you can have the, primary structure representing the arrangement of the amino acid one next to the other. You can make out these amino acids join through the peptide bond. And this particular primary structure can take up the form of the coil or may take up the form of the folding form. Though they fold and they can coil here. And this particular rigidity is going to be, or they are going to be stable in the coiled form or in the folded form through the formation of the hydrogen bonds here. So hydrogen bonds, although they are going to be very weaker, but because of an extensive hydrogen bond being existing within the alpha helical structure as well as the beta plated sheet, they actually give strength to the structure here. So then coming to the triple helical structure. So such, it is going to be what is called as the thread-like structure. So you can make out three helix, triple helix. That's why it is called as a triple helix here. So this triple helix is going to be like the, it appears as a chain of a rope, which forms a rope here. Okay, so which is going to be involving an repeated arrangement of the glycine with hydroxyproline as well as the hydroxylysine. So the next level of the organization is going to be nothing but the tertiary structure here. So tertiary structure is very important because this tertiary structure gives function. They are going to be responsible. They are responsible for the functioning of the proteins here. So there are numerous proteins are there. Yeah, this is the protein, tertiary structure of the proteins here. Some proteins act as transport molecules, some of them as structural molecules, some are acting as the enzymes here, some are going to be acting as the hormones here. So varied functions are going to be given. How they are given, and that is been through their arrangement, their foldings, their conformation, their arrangement within the 3D space here. And this tertiary structure is going to be stabilized by the numerous bonds here, like as you have seen the hydrogen bonds, no doubt they'll be there, but along with that you will also be having what is called as the disulfide bonds here. Disulfide means di means two, sulfide means this cysteines are going to be the sulfur containing amino acid. This are going to be joined through the disulfide bond, which are actually going to be covalent in nature. So along with that, you will be having an hydrophobic interaction. Hydrophobic interaction means the one which do not actually like water. So if the R group, if the side chain of the amino acids are going to be hydrophobic in nature, so hydrophobic side chains are going to interact with respect to each other, as well as you can also get this hydrophilic interaction. So numerous of such compounds will give rise to the tertiary level of organization or the tertiary structure of the proteins here. So tertiary structure of the polypeptide chain, as I told you, they are going to be giving this particular function to the compound. So similar polypeptides, if they are going to be brought with respect to each other, giving rise to a highly complex structure, which you can observe in case of the hemoglobin here. There are actually 4 polypeptide chains are brought together. They may be actually, two similar polypeptide chain can come together, or maybe different polypeptide chain also can come together to give rise to a complex structure. Okay, then this is actually not there in the syllabus, but it's very easy to understand. This is actually is an Anfinsen experiment where the ribonuclease A is the enzyme here. I will take another 2 more minutes. So ribonuclease A is going to be the enzyme involved in breaking of the RNA molecules here. So naturally, the function is due to the its conformation. Okay, 3D structure, native structure. But when the urea and mercaptoethanol is going to be added to that, which are going to be toxins, so they are going to break all those stabilizing interactions like the hydrophobic interaction, hydrophilic interaction, disulfide bonds. All these are going to be broken down except but for the peptide linkages. Peptide linkages are intact, but other than that, every other linkages are going to be broken down at the when you are going to be adding the toxic molecule. Okay, change in the heat or the pH values, all these are going to be breaking the interactions which are going to stabilize the tertiary structure. Naturally, when it is happening like that, they are going to be losing their function. So ribonuclease A will lose its function of breaking the RNA molecule. That is what is called as denaturation. Okay, they lose their activity. But remove that particular, urea and mercaptoethanol. So naturally, the disulfide bonds are going to be regained, or it is going to be reformed, giving back the activity. So these are some of the questions I thought I will just ask you. So which functional group participates in the disulfide bond? I told it cysteine. A nanopeptide contain how many peptide? Nano means 9 amino acids will be there. You will have 8 peptide linkage will be there. The artificial silk. So silk is involving a fibrion here. Fibrion is a beta pleated sheet. Okay, it is going to be having that secondary structure, but artificial silk is not going to be having a peptide linkage because it is not a protein here. Okay, amino acid is a proline. Proline is an I think you saw it under the neutral amino acid here. So histidine is the this is actually the structure of the proline. Proline you can make out this is going to be non-polar part here. Okay, histidine on decarboxylation forms histamine here. They are going to be involved in the allergic responses. Okay, this is the structure of the histidine here. You remove this carboxyl group, you get the histamines. Okay, that's why you take antihistamines whenever you are going to be having an allergic reaction. So these are the two questions which Shri Siddhegowda will be sending to you. Thank you so much. I think let me stop sharing.
Yes, madam. I already shared to chat box.
Okay. Thank you so much. And thank you again for giving me an opportunity to present. Thank you, KSTA, thank you, Dr. Ramesh, and thank you, Sri Siddhegowda, and all the faculty members who are involved in this particular program. And thank you, the attendees, students. KSTA is highly appreciable in initiating this program to help you out people. Thank you so much, all of you.
Thank you a lot, ma'am.
Thank you so much.
ಸೊ ಇಂದಿನ ಪ್ರವಚನ ಕಾರ್ಯಕ್ರಮದಲ್ಲಿ ಬಯೋಮಾಲಿಕ್ಯೂಲ್ಸ್ ವಿಷಯದ ಮೇಲೆ ತಾವು ಸರಳವಾಗಿ ಸುಲಲಿತವಾಗಿ ವಿದ್ಯಾರ್ಥಿಗಳಿಗೆ ಅರ್ಥವಾಗುವ ರೀತಿಯಲ್ಲಿ ತಾವು ಕೊಟ್ಟಂತ ಉಪನ್ಯಾಸ ಕಾರ್ಯಕ್ರಮ ಭಾರಿ ಅಚ್ಚುಕಟ್ಟಾಗಿತ್ತು ಎಂದು ಹೇಳುತ್ತಾ ತಮಗೆ ನಮ್ಮ ಅಕಾಡೆಮಿ ವತಿಯಿಂದ ಧನ್ಯವಾದಗಳನ್ನು ತಿಳಿಸುತ್ತಿದ್ದೇವೆ. ಹಾಗೂ ನನ್ನ ವೈಯಕ್ತಿಕವಾಗಿ ಧನ್ಯವಾದಗಳು.
ಥ್ಯಾಂಕ್ ಯು ಸೋ ಮಚ್. ಥ್ಯಾಂಕ್ ಯು.
ಹಾಗೂ ಈ ಒಂದು ಕಾರ್ಯಕ್ರಮಕ್ಕೆ ಪ್ರತಿದಿನ ಶ್ರಮಿಸುತ್ತಿರುವ ಅವರ ಅಕಾಡೆಮಿಯ ಮುಖ್ಯ ಕಾರ್ಯನಿರ್ವಹಣಾಧಿಕಾರಿಯಾದಂತಹ ಡಾಕ್ಟರ್ ಎಂ ರಮೇಶ್ ಸರ್ಗೂ ಕೂಡ ಧನ್ಯವಾದಗಳು ಸರ್. ಹಾಗೂ ಅಕಾಡೆಮಿ ಅಧ್ಯಕ್ಷರಾದಂತಹ ಡಾಕ್ಟರ್ ಅಯ್ಯಪ್ಪನ್ ಸರ್ಗೂ ಕೂಡ ಧನ್ಯವಾದಗಳನ್ನು ತಿಳಿಸುತ್ತಾ ನೆರೆದಿರುವ ಎಲ್ಲ ನನ್ನ ವಿದ್ಯಾರ್ಥಿ ಮಿತ್ರರಿಗೂ, ನನ್ನ ಸಹೋದ್ಯೋಗಿ ಮಿತ್ರರಿಗೂ ಹಾಗೂ ವಿವೆಕ್ಸ್ ಗ್ರೂಪ್ ಮಿತ್ರರಿಗೂ ಕೂಡ ಧನ್ಯವಾದಗಳನ್ನು ತಿಳಿಸುತ್ತಾ ನಾಳಿನ ಉಪನ್ಯಾಸ ಕಾರ್ಯಕ್ರಮದಲ್ಲಿ ಡಾಕ್ಟರ್ ನವೀನ್ ಸರ್ ಅವರು ಆಲ್ಟರ್ನೇಟಿಂಗ್ ಕರೆಂಟ್ ಎಂಬ ವಿಷಯದ ಮೇಲೆ ಉಪನ್ಯಾಸ ನೀಡಲಿದ್ದು ವಿದ್ಯಾರ್ಥಿಗಳೆಲ್ಲರೂ ಇಂದಿನಂತೆ ನಾಳೆನೂ ಕೂಡ ಹೆಚ್ಚಿನ ಸಂಖ್ಯೆಯಲ್ಲಿ ಸೇರಬೇಕೆಂದು ಮತ್ತೊಮ್ಮೆ ಸೂಚಿಸುತ್ತಿದ್ದೇನೆ. ಥ್ಯಾಂಕ್ ಯು ಮೇಡಮ್ ಒನ್ಸ್ ಅಗೇನ್.
Thank you so much. All the best to students.
Thank you.